Hi, I'm Dr. Karen Goldberg, and I'll be giving you a tour today of what happens to an eDNA sample once it gets to the lab here at Washington State University. One thing that all eDNA labs have in common is that we work hard to keep our high-quality DNA that we might be using in a tissue lab or a lab where we process PCR products away from our low-quality or low-quantity eDNA samples. So this is a dedicated room where we have to shower and change clothes into clean clothes before we come in if we've been in a lab with tissue samples or a PCR product. So let's go. So the first thing that happens when we come into the lab is that we clean the whole lab bench down with 10% bleach. This whole lab gets cleaned with bleach regularly, as well as all the pipetters being UV sterilized and various other clean room practices. So this is our sample, and the first thing that we do with the sample is to divide it in half, and that's to make sure that if something goes wrong or we have some question with the results, we still have an original half to go back to confirm our findings. Now I have my tools set up here, tweezers and a scalpel to divide the sample in half. And then I've got two tubes waiting, one of them for my extraction negative and one of them for this filter to digest it. So we bring the filter out onto a clean surface. These implements have been cleaned in 50% bleach and then rinsed in water to get rid of the bleach residue. So the half I'm saving goes immediately back in the envelope. Now this envelope's been out in the field, may have touched some other things, so now that I've handled it some more, I'm going to change my gloves. And then I'm going to divide the filter up into enough pieces so that it can squish down into the digestion buffer and get fully digested. So we usually use about 4 pieces. You can see as we squish it down that the digestion buffer has bubbled up over it so that we know we have enough. So the next thing that happens is that these samples get mixed in the vortex, And we do this several times during overnight digestion. So for the rest of the day, we'll be coming in and occasionally putting these on the vortex to make sure that all the DNA in the filter is getting to experience the digestion. So after the samples have had a chance to digest overnight, we use these little columns that separate things out and also kind of molecularly separate things out, which can help increase our detections of eDNA in the sample. So now we use the centrifuge to separate the liquid that has the DNA in it from the filter. After many more steps of the extraction process, we've now purified the DNA and gotten it hopefully away from all the other impurities in the sample. And in the end, we get our samples in these tubes and we put them in the fridge for later analysis. So when we're ready to analyze samples, we use quantitative PCR, so we have a quantitative PCR plate here, and we load the samples in triplicate wells, so that each sample is analyzed 3 times. So after adding standards and sealing the plate, we bring it up to be analyzed in the thermocycler. This machine will heat and cool the DNA sample with the enzymes and make the PCR reaction go. From this process, we'll get millions of copies of what we're looking for, or in the case of a negative sample, we'll get no copies. And each one will glow at a certain amount, which will enable us to quantify how much DNA was in the sample. So after thermocycling is complete, this is what the results end up looking like. And from these results, we can tell whether we detected the DNA of a species in the sample, as well as how much DNA of the species was in that sample.